We're now going to move into talking about diffusion imaging. Diffusion imaging is a way to look at the white matter structure of the brain, but we can also use it to understand microstructure of gray matter regions. Diffusion imaging is based on random motion of water molecules through tissue and on a scan. So just we see here, this is an example of diffusion imaging. We have this area that appears dark, and that is part of the lateral ventricles that are filled with cerebrospinal fluid. And so where water molecules and the motion of this water in the brain is really unrestricted, we see it appearing dark on this type of imaging. Now this is going back to our introduction at the beginning of the semester, and so hopefully some of these slides are familiar. If not, then this is a refresher. But diffusion imaging really dates back to the 1800s to a botanist named Robert Brown. And he was doing some experiments, and he had a cup of water and had pollen that was on the top, on the surface of the water. And he looked at it closely and saw that the pollen was moving around. It jiggled as if it was alive. And so he started thinking about it and thinking, is there this inherent life in the pollen? And but went on to describe this, what we call diffusion. And so molecules, though, are really in constant motion at non-zero absolute temperatures. And so there's never really still. And this is what Robert Brown really was one of the first to mention, to talk about and notice. Then Albert Einstein came along and put a mathematical model to the diffusion. And so he was able to describe it mathematically, this principle that Robert Brown had noticed qualitatively. And we won't go into all of this, but again, this is the model that Einstein had come up with and described. Essentially what it means, and what Einstein had described, is if you're in some medium, you don't have barriers. So you could think of this as within the ventricles in the brain, where you're right in the middle of it, surrounded by water. The diffusion displacement is going to follow a normal distribution, this Gaussian distribution. And it'll take, in some given time, the water will displace in that a certain distance. And we won't get into all of that in this class of how to calculate that. We'll let our software do that. But that's what is going to be calculated behind the scenes, is how quickly the water molecules are moving, where they're going, and by where, we'll say it's really kind of a non-specific where, but it's just, are they moving in some particular direction? Meaning, are they kind of going left, right, front, back, up, down in the brain? We can't really describe where they're starting, where they're ending up, but we can trace the movement of water. So why is diffusion of water in the brain interesting? What does it tell us? So when we are in areas that are open, like the ventricles that are filled with cerebrospinal fluid, there's nothing really to restrict the movement of water. And so that's this isotropic, free movement of water that would go out in a sphere. But when you get to the tissue boundary, you have the membranes, the walls of the cells. It could be something pathological. Again, it could be tumor. It could be whatever it is in the brain. Then the diffusion of water will be somewhat restricted. And so an example here with a neuron, so the cell body, water movement is relatively unrestricted in there. If we're tracing our molecules, they can bounce around within the cell body, water is small enough it can go across the wall. There might be a little bit of restriction there with the walls of the body, the cell body, but in general it's going to be quite unrestricted within the body of the neuron. But as we are in the axon, particularly in areas where there's myelination, and these are formed by glial cells, then the water will tend to diffuse along the length of the axon. Of course, some can escape. We have these little open areas, nodes of Ranvier, as they're called, and water can get across the membranes of the axon, the myelination that's going around the axon, but generally there's going to be more restriction of diffusion out than there is up and down the axons. And so that can tell us about structure in the brain and what we call the microstructure in the brain. So again, just another way to think about it, unrestricted diffusion within cell bodies. And so this is, we'll come to these a little bit later, but this is what we call an ellipsoid. And this is representing our diffusion in 3 dimensions. So in the cell bodies, in cerebrospinal fluid, we're going to have our more spherical diffusion. Kind of looks like a happy face. It's supposed to be a sphere. But that would be our CSF. And then within cell bodies, little restriction, but relatively unrestricted. But then when you're out in the white matter, and these are axons, especially axons that are bundled tightly together in these white matter pathways, these tracks, then the diffusion will tend to follow down the axons. And so the displacement will be very linear. And so what we can do again is measure this diffusion, and so then there's a term that we call the apparent diffusion coefficient. And so that really tells us, that's a metric that tells us, is the diffusion relatively free, unrestricted, or is it restricted? And so that's going to be one thing that we calculate when we're processing some diffusion data. So diffusion MRI is, one way to think about it is it's kind of a form of T2 imaging. We haven't talked a lot about T2 imaging, but if we look at this image here, so it's not exactly a T2 image, but pretty close, but the CSF is bright. There's no diffusion encoding on this. We use this as sort of a reference scan. And then everywhere else is darker. White matter would be dark. Gray matter is a little less dark. And then, of course, cerebrospinal fluid is brighter. And so we get this reference image. And then we acquire multiple directions, what we call gradients. And the gradients are going to be applied in different directions. And so we can think about that as looking at ways to plot our 3-dimensional space, and we're sampling in multiple directions. So we're going, and we can think about it. So here, this is showing 6 images. So if we want to have 3-dimensional space, and we can represent it here with these axes, we have our X, our Y, and our Z coordinate system. What we're trying to do is get an overall picture of where the diffusion of water is, where the water is going, how the diffusion is happening, how quickly it's happening. And so what we want to do is sample as many different directions. And so at a core, we could sample the y-axis in a positive direction. We could sample it in a negative direction. Same thing with X, and same thing with Z. So we're going to sample all of these. And I guess I should mark that one as our negative and our positive be coming out of the out of the screen here. But so we'll have our different directions, and so that, again, that's our minimum. We can look one time in each direction along the major axes, and so that's our 6 gradients there. But just like any sampling, we'll tend to get a more accurate picture for sampling more. And so 6 directions is what is kind of the minimum that we would look at. But the recommendation would be 6 times or 7 or 8 or 9 or 10 times that we're going to sample, and sometimes even much more than that. So that gives us a very fine picture of where the water is going. So again, what it can look like is if we sample in one direction, then we can start to see the structure of white matter in particular, because that's where diffusion is most restricted. And so here we're sampling in the X direction, and then we're seeing here these are pathways that are going primarily, water that's traveling primarily front back in the brain. We're sampling in the Y direction now. What's being highlighted, where we have the restriction, it would be the water that's moving in and out, really in the Z direction. We're sampling in Y, but what we're seeing is movement in, what's being highlighted is movement that's in an orthogonal plane to that. And so then that's moving in and out, up and down in the brain. And then over here, sampling in the Z direction. But here, what's being highlighted is movement really left and right in the brain. And as we can see, if we're looking throughout the white matter, in here there's not a lot of left and right. There's some, certainly. There would be some up here, not quite as strong as this particular area. But here at corpus callosum is where we're getting a lot of this left-right movement. And so again, more directions we apply these gradients, then we can start to get a clearer picture of the structure of white matter at any spot in the brain. So where's the white matter there going? Where's it going there? Where's it there? All over the brain. So again, as a reminder, we acquire the picture of the brain that in the MRI we acquire in slices. And it can be in one plane, so this is a coronal slice. And so this would be like looking somebody in the face. So eyes would be, it would be down here, kind of in front of this. We're farther back. This is brainstem right here. That's the pons. These are our ventricles. And here's white matter. These are white matter pathways, corpus callosum right there. But we acquire slices of the brain, and then it can be reconstruction in three dimensions. So most diffusion imaging currently is about a 2 by 2 by 2 millimeter voxel. So that's 8 millimeters cubed for a volume. And that's relatively high resolution, but there can be hundreds, tens to hundreds of thousands of axons within that area, many cells. So really kind of a low resolution overall in the brain. So that's what we're really acquiring are voxels in each spot, each pixel. It's a volumetric pixel. And so we want to understand for each voxel, what does diffusion of water look like? So we can do that within white matter and gray matter. We really want to understand, is this gray matter, is it white matter, is one of our first questions. And historically, diffusion imaging primarily looked at white matter, but we're also applying it to understanding gray matter structure. But if this is white matter, what pathway is it part of? So gray matter or cell bodies, white matter would be connections. These are the pathways. And that's really the power of diffusion imaging, is understanding the structure. So what's the orientation of the diffusion? What's the magnitude? So what direction does it look like the water is going? how quickly is it getting there? Again, is it on an interstate, or is it on some small local road where there's a lot of traffic, or that's very narrow and not a lot of movement? We can't capture all of that in a grayscale image, and that's where having color helps. So color here, red can be more left and right, and green up and down in the brain, and then blue. So again, red left and right, and then blue would be in and out, kind of front to back in the brain with that color scheme. So what is the diffusion tensor? So that is one of the simplest ways to characterize the movement of water in the brain. So again, if we remember, I had drawn the axes. The axis didn't quite meet up. So we have our three axes, and so a tensor model is a way to orient diffusion in space, describe it in three-dimensional space, x, y, z. And so we can characterize diffusion really where the diffusion of water isn't on one plane, on one axis. It doesn't have to be just there on that axis. We can understand the movement of water if it's maybe going in some direction, kind of like that. And so a tensor allows us to get the mathematical representation of water diffusion in space. So the elements of the diffusion tensor, it's a symmetric 3x3 matrix, and it has 6 unknowns. So symmetric, so these values right here are just repeating of these off diagonal values. So here's our diagonal values, and they're off diagonal values. And so these ones are repeats, and because we have 6 values, we need to have at least 6 directions to solve the equation. This is based off of linear algebra. And so the diagonal elements are proportional to diffusion displacement. That's the apparent diffusion coefficients along 3 directions. So those are 3 primary directions, again like x, y, z. And then we get our off diagonal elements are going to be modeled by the rest of the matrix, the other 3 values. So what this can look like is if we have zeros off diagonal, that means the water is moving in what we call an isotropic diffusion. It's spherical. The magnitude of these values will indicate, that's the apparent diffusion coefficient, that's how fast is the water moving, essentially. Is this a little trickle of stream, or is it a fast moving torrential river? And so that's telling the difference. So again, if we have these off diagonal zeros, then it's perfectly isotropic. That's pretty rare in real life, but that would be the ideal. And if they're not all exact same in magnitude, then we start to have a little bit of shape here, different than our sphere. So now we're getting more of a potato or a football shape, and so that has more movement in one direction than the others. And again, if this 6 was here in the middle, then we would have a little bit more movement in the y direction. Or if we had a 6 and then these were both 3s, then it would be z direction. And so that's how we can start to represent water movement in the brain. And again, the magnitude and really the ratio between your primary, your largest value on the diagonal, and the other two will tell you the shape. So if you have a really high ratio, this 8:2, then it's really cigar shaped in this case. We're moving into pencil shape. And then now if we start to have some non-zero off diagonal values, again, see here's the symmetry. Those are the same value. Then it has some other orientation space. It's not lined up exactly on the XYZ planes. So again, this is going to be more realistic in the brain, where the values of the diffusion isn't going to be exactly in plane with the gradients. And so what you can do is start to describe this matrix by the size of the principal axes. Those are your eigenvalues. So here's our lambda 1, 2, and 3. And then the direction of the principal axes would be an eigenvector. And so we have eigenvalues and eigenvectors. So again, the eigenvalue is going to be equal essentially to what is that value along the primary axis of the matrix. We have our diagonal and we have our off value. We have our off diagonal values, and so these eigenvalues will tell us the magnitude of diffusion. So the tensor matrix, again, that looks like this. Here are our eigenvalues, lambda 1, 2, and 3. And lambda 1 indicates the value of the maximum diffusivity, or that's our largest eigenvalue. So what you do is you identify that first. I mean, you do the mathematical reconstruction for every voxel in the brain. So again, we have our little cubes. They don't have to be cubes if it's not, say, like a 2 by 2 by 2. Sometimes you get a 2 by 2 by 10 or a 2 by 3 by 8. Again, the shape of this can be different, but the ideal is a cube, and so most of the research quality data we're going to be acquiring it, at least recently within the past 10 years or so, is going to be acquired with what we call isovoxel, where it's the same size on each direction. And so what we do though is, as we're solving our linear equation using linear algebra, we're going to solve our matrix. We're going to figure out what's a primary eigenvalue, and that would go up in the upper left corner. And so that's your magnitude, primary diffusivity, and so where is it going? And then lambda 2 and 3 are defined relative to lambda 1, and they're set up to be orthogonal. So again, if lambda 1, let's say we have some magnitude and some direction, was right there, so we get our lambda 1, then lambda 2, so again, here, let's say our magnitude here is 3, this is a 2.5, and then we're going to get our lambda, that's our lambda 2, and then we're going to get our lambda 3, and I'm just going to draw it up like that. That's orthogonal. They're supposed to be orthogonal, and maybe that magnitude is only a 1. But we define lambda 1 first, and then set lambda 2 and lambda 3 to be at 90-degree angles from lambda 1 and each other. So we end up with a 90-degree angles here. So this would be 90 degrees between 1 and 2, 90 degrees between 3 and 2, and 3 and 1. And so that's kind of important if you go on to delve more technicality with diffusion imaging. You want to understand that, because if your estimates of lambda 1 are off a little bit, and they could be off because of pathology in the brain, then what you end up with is also incorrect lambda 2 and 3. And with that, that's very technical, we won't go into that, but it's something to be aware of. There are imperfections with how we're modeling water diffusion in the brain. And again, just to show it, if lambda 1, 2, and 3, sorry for the yellow, kind of hard to see, if they're all about equal, that means diffusion is isotropic. If one is greater than the others, then it would be anisotropic, meaning there's some directionality to it. So what we can do is we can put somebody in the scanner and get an image of their brain. This is a T1 image, and so our white matter in here is white, but we look at it, we don't have any understanding really of the structure of it. We don't know what this is doing, because the white matter are pathways, these are connections, and we can't see where this is connecting or anything about it. But with diffusion imaging, we can look at the direction of the eigenvector, and so the direction of the greatest eigenvector is going to be whatever it's associated with the primary eigenvalue. And then what we can do is calculate a tensor at each voxel, and that allows us to draw a little line, in this case, to represent what is the white matter looking like at that voxel. What's the direction of the displacement? We don't really see necessarily the magnitude with this image, but we are seeing these are our eigenvectors, and so we do see direction. So now we look at this image and we don't see any structure over here, but if we look over the diffusion image, now we can see, now we have all these eigenvalue, the eigenvectors associated with the primary eigenvalue. All of these are going up, and they keep going up, and then it looks like there might be a turn here, or we also have a turn in that location, maybe some fibers that are crossing each other that it's not really clear in there. And so we start to get an understanding of maybe what is the white matter doing. So that's the benefit of diffusion tensor imaging, is we can reconstruct displacement of water in 3 dimensions. We can also color code it, and then this color coding is based off of the eigenvalue and the eigenvector. And so red, primary left and right, blue up and down in this case, and green would be front to back. And some of this we kind of need to ignore because there's a lot of, some of this is front to back, but some of this is also within structures. So right here we have a caudate nucleus, and that's a gray matter structure. There is some white matter that goes in and out of it, but again, it's kind of showing up as green in here just because there's a lot of complexity in there of where the water is going, meaning it's going in every direction. We don't really have a good idea. So color isn't always the best way to look at it, but certainly for major pathways, we can get an idea about the structure. So now we want to have some way to, through research, we want to quantify this diffusion. So the simplest method is what we call mean diffusivity. So the mean diffusivity is the mean of the 3 lambda values, so our 3 eigenvalues, lambda 1, 2, and 3. And so this is an inverse measure of membrane density. It can be similar for gray matter and white matter, and it's higher for CSF. And so that's why on this image we see white, bright signal, meaning very high mean diffusivity. That means there's not a lot of restriction in where or how fast the water is going. But everywhere else in the brain there's some restriction. This is sensitive to what we call cellularity, edema, and necrosis. So clinically, the radiologist would look at this type of scan to look for evidence of tumor, evidence of cell death, of swelling, anything like that could show up on here. So again, if we had a bright spot in here, because I could change the color. But if we had someone came in for a scan and they had areas within white matter that were more bright, that's unexpected, and so that could indicate increased diffusion, higher mean diffusivity, and that isn't necessarily good in that case. Another index of diffusion is what we call fractional anisotropy, and here's the equation for it. It's a very useful value to describe movement of water, really, so as the directionality of it. The range is between 0 and 1, values close to 0. The diffusion is spherical, the isotropic diffusion. And values that are close to 1 are going to be more tubular. They're going to be more shaped like a pencil. So this doesn't tell us where it's going, what direction it's facing. It's just, is the water displacement directional? Is the shape more like a cigar or more like a sphere? So this is a value that's probably used the most in all of diffusion work. There are issues with it. We won't really get into that in this class, but the more and more research is done, there's lots of research that shows it's useful, but there's a lot of new research that's showing that there's a lot of limitations, and we're trying to move beyond that value. You have other summary measures, what we call axial diffusivity. That's the greatest of the 3 eigenvalues. Again, that's just our lambda 1. We also have radial diffusivity. That's the mean of lambdas 2 and 3. Radial diffusivity, the assumption is that it can tell us about the integrity of myelin. So if we think about it, if we have our axons and we have our myelin that's wrapping around, so our primary diffusion in this case, lambda 1, is going to be up and down the axon. Remember, that's defined first, and so that's lambda 1, but again we also have a vector associated with that, and then we define lambda 2, lambda 3, and so lambda 2 and 3 should be going across the myelin. Remember, this is 3-dimensional space. And so the assumption is that that can tell us about integrity of myelin. So if you have higher radial diffusivity, if lambda 2 and 3 are higher, then that might indicate that your myelin has either disappeared or part of it is broken down. There's some alteration to it. Research suggests that that's a pretty safe assumption, but there are issues with it we won't go into for this case, for our class. And in this instance, we'll just leave it at that. The radial diffusivity might tell us something about the structure of myelin. So again, just a summary of what the images look like. Our fractional anisotropy image, mean diffusivity, longitudinal ADC, so that's also called, that's our axial diffusivity. That's our apparent diffusion coefficient. So it's longitudinal ADC, called primary ADC. There's a lot of terms for it. And then transverse, or also called a radial. And so we can see the difference in what the images look like. We can see areas here that are dark. So if they're dark, what does that mean? That means there is lower transverse ADC, the lower radial diffusivity. So these areas of the corpus callosum, very tightly packed white matter, myelinated, going left and right here in the middle of the corpus callosum. So these are fibers that are almost all unidirectional, and so there's not a lot of diffusion out in the other directions. So low diffusivity. So what we can do is use these values, these metrics, to tell us something about the structure of the brain. Fractional anisotropy will decrease in some cases, and when it does, there's typically a mean diffusivity increase. And so again, if FA decreases, FA is a value between 0 and 1. So if FA decreases, mean diffusivity, which is the average of those 3 eigenvalues, so mean diffusivity tends to go up in areas where FA declines. And so when you have, particularly when you have both of those, FA decreasing, mean diffusivity increasing, that tends to indicate some sort of breakdown in tissue. We can see fractional anisotropy changes in multiple sclerosis. Even in white matter that might be what's called normal appearing white matter. So if you do a scan, you look at it with the T1 image, you might not have any evidence that there's something wrong, but if you do a diffusion scan and you calculate fractional anisotropy in those regions, we can see a slight decrease. So it can be very sensitive to changes, alterations in tissue in the microstructure. And again, you get changes with mean diffusivity. These are different types of multiple sclerosis, normalized pixel count. Again, so there are different ways to compare the diffusivity between groups. This is a little bit older method. It still can be done, but we have newer methods as well. So we can put all this together, and just for nice pictures, these aren't particularly helpful for research, but we can get our colored FA images, these maps that'll show directionality. And they're pretty pictures, but again, don't tell us a lot. So if we have a quantitative diffusion map, so that's, we calculate fractional anisotropy, our parent diffusion coefficients, we get all of these standard metrics. What's the cause of it? So if we have a FA decrease in white matter, it can be caused by a decrease in our lambda 1, and that might indicate a break in the axon. So again, lambda 1 tends to be in line with the axon, so if lambda 1 decreases, maybe there is some break there and some axons are part of them. It could be due to an increase in transverse apparent diffusion coefficient. So maybe lambda 1 isn't too bad. That's still pretty intact, but what we've done, instead of having our nice myelin everywhere, we start to get a breakdown of myelin. And with a breakdown of myelin, we can get more diffusion out in these other directions. So we still have diffusion up and down the axon, but again, some of these molecules, they get to these areas where there's a breakdown and they can more easily cross and get out in these other directions. So just because we have a different value in FA doesn't mean we know what's going on within the brain. And we can't overinterpret results. We always have to be very careful in what things can mean. And diffusion tensor imaging is an oversimplification of reality. We'll get to some of that later. So again, here's another way to look at it. So if this is normal, let's say this is normal for a patient, then you can have an increased FA, decreased mean diffusivity. That might just be some sort of swelling. We get swelling of the cells within the area. We could have increased FA, lower mean diffusivity, and have higher density. So again, swelling could be bad. Higher density might be good, meaning someone has high FA, low mean diffusivity, just because they happen to have many more axons in an area. Or there could be swelling. So again, we don't always know the etiology of of what this means. You could have a decreased FA and increased mean diffusivity with loss of myelin. So all the cell bodies are there, the axons are there, you just lost the myelin. We'll see a change that could look like that. Or we have nicely myelinated axons, but we've lost some of the cells, we've lost some of the axons, and so the metrics could look the same. So again, just because we have higher FA in one group or over time, or lower FA in one group or over time, doesn't mean we know what's causing it. But as I said, the tensor is really an oversimplification of what white matter looks like and what the water is doing in the brain. And so there's what we call high angular resolution diffusion imaging. HARDI is our acronym for it. And this allows for more complex mathematical models to look at the microarchitecture. So in an area where we have crossing fibers, the diffusion tensor analysis would look like this. It would be this ellipsoid. It would look like almost like there isn't a lot of structure to the white matter, to this area. It could almost be like these are cell bodies. This is gray matter. But that's not the case at all. It's just crossing fibers that are giving very strong signals in each direction. And so that washes out with diffusion tensor imaging. And again here, we can see sort of a watermelon shape. In this case, we also will have crossing fibers, but instead of being at closer to 90-degree angles, they're at some other angle. Again, they could be crossing like that. Or it's also possible that these fibers are curving. That's kind of too sharp of a curve. That's not, it depends, you know, there's an angle to this curve, and we don't have really sharp angles in biological features in our brains, and so we don't know if this is a curve. This is probably crossing fibers. But in areas that's very directional, this HARDI method really isn't any better than diffusion tensor imaging. But again, more complex white matter, which some estimates say is about 90% of the brain, is going to be complex, more like this, rather than that. Something that looks like that might only be about 10% of the brain, of the white matter. So HARDI methods are going to be much preferable. But again, for our class, we're just going to stick with our classic diffusion tensor imaging. It's still widely used and useful. So remember, a tensor has 6 unique parameters. So to estimate those 6 parameters, we have to acquire at least 6 diffusion weighted images. Recommendation is about 21 will give us good stable data. HARDI models have more parameters per voxel, and so you have to acquire more images. This is saying more than 30. You can do it with about 30, but really recommendation is going to be at least 48 directions that you have to acquire. Greater than 60 is going to be recommended, but 48 should give you enough. So we can take those reconstructions of displacement of water, this diffusion within each voxel, and start to do tractography. We do fiber tracking. We won't go to this video, but again, we can start to draw those lines as I had done on a previous screen to try and connect white matter pathways. So again, we might have pathways there, we might have pathway there. This dark, this is diffusion, displacement in kind of a spherical shape. Again, so we can't really know where that's going, but some of these other ones we can maybe draw lines like that, and then we can start to draw these lines and do fiber tracking. And so again, that's called streamline tractography. It's a what we call a deterministic method, because where the fiber goes next is determined based off of the underlying structure. And so this is pointing in that direction. We would set some rule saying it will go in one direction unless it has a turn of greater than some degrees. 50 degrees, 60 degrees, 70 degrees, somewhere in that range. Typically 50 to 60 degrees is used as a cutoff. If it's higher than that, we won't draw any connections. If it's lower than that, so here we could draw a connection, and there we draw a connection. So again, we can get some information about multiple orientations of fibers. Another method that we will use in our class is probabilistic tractography. We'll actually use a combination of both of these, the streamline and probabilistic, but most of what we're going to do is this probabilistic tractography, and so then that will just give a probability of which direction is it going, and it gives a lot more range in where it can connect. There's a lot of unknowns in the data, and probabilistic tends to be pretty sensitive, sometimes maybe a little too sensitive, meaning it's sensitive but not very specific. And we'll skip over that. But again, deterministic, the streamline tractography, we draw a line, we connect the dots, essentially, based off of a rule. Probabilistic, this really provides more tracking into complex regions, although there are deterministic methods that use very complex mathematical models that can do a similar thing. And it also provides this quantitative probability of connection at each voxel. So I had drawn earlier, there are instances where we have a lot of crossing fibers, where we have fibers that appear to kiss, and they just touch. And within those areas, right where that intersection is, we're going to have low fractional anisotropy. So remember, FA ranges from 0 to 1, and 1 we assume is very directional. 0 is going to be isotropic. 1 is anisotropic. And so 0 is it's going out in every direction equally. And 1 would be perfectly linear. So in the brain, we're not ever 0 and not ever 1. We can get close to 1, but in these areas where you're crossing or kissing fibers, FA is going to look lower. So instead of being high, which it should be because we do have very directional fibers, it's very clear that we have these clear pathways, but what happens is they sort of cancel the signal of each other out. And so we're not really getting to see the structure when we use FA as a metric. So what that can look like in the brain, so there's the area here in what we call frontal forceps. It's just white matter, where we have a lot of white matter coming out of corpus callosum and meeting up with other major pathways. FA in that area is really low. So here's areas where FA is high, but it looks dark, sort of dirty in there. And so that's a low FA. And that's a problem. That doesn't mean, normally we'll think low FA, that's bad, but we could have low FA because it's really good. We have really strong crossing fibers. So what the Hardy method is, it allows us to see crossing fibers. We'll skip over this quickly. But that same area in the brain, so here's corpus callosum. Again, we have these very linear models, but as we're getting out here, we start to see all of these crossing fibers. And so we can have low FA in that region. FA, remember, is based off of diffusion tensor imaging, and it doesn't really allow us to look at crossing fibers. It just picks one direction, or we could have lambda 1 and lambda 2 are pretty close to one another in size and magnitude, and that can be okay, but that doesn't really tell us much about the structure. And so as we're going on to calculating fractional anisotropy and some of these other metrics, they're going to be thrown off by that. Again, zooming in, we can see that there's a lot of crossing, and in some places multiple directions. Here, 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 we can have 3 or 4 or 5 different directions within these areas. It's relatively rare to have that many different directions, but 2 to 3 to 4 can be pretty common. So what this can look like, so this is using diffusion tensor imaging. This is the brain stem. This is cortical spinal tract. This is the pons. The front of the brain would be that way, the eyes. And this is middle cerebellar peduncle. If you've had neuroanatomy, might recognize that term. But this is white matter that's going in and out of one cerebellar hemisphere and kind of wrapping around the pons, the front part of the brainstem, the little bulge in the front, and going around into the other side. And with DTI tracking, what we see, we have cortical spinal tract going up. There's still some cortical spinal tract going up, but we get a lot of fibers that look like they're leaving the cerebellum and then going up. And that's not really what's going on. So with the Hardy method, we can see, now we can see that the fibers go through, and we have these 2 clear major pathways. I'm going to go again. This Hardy method provides a lot more resolution, really, to what's going on in the brain. So some of the things we'll be dealing with, that's all background, but for our class for the processing, what we're going to be dealing with are problems in the data. We won't give you really problematic data, but we'll have to correct for this. So when we acquire a scan, this diffusion imaging, the sequence is called an EPI sequence, and it's related to what we do for a functional MRI scan. But this is really susceptible to tissue and air interfaces. So this is the front of the brain, and unless you've had some major head trauma or a stroke or something, the front of your brain doesn't look concave like that. It really should be out convex like that. But what happens is the signal is either lost or it's displaced inward, and that's due to, in part, to these sinuses up in the skull that are filled with air or could be infected and filled with that infection. But the sinuses can distort the signal really on some of the edges of the brain. We get some distortion back here as well. And so this warps, that area can warp the entire image. And some of that can be dependent on how good the scanner is, but some of it is just your anatomy. And so what we want to do is have a way to correct for that. We can also get eddy currents as you are measuring water diffusion in one direction. Again, that's the scanner, the head coil as we call it, puts a radio frequency signal into the brain, and that aligns protons along with that signal, and it waits to measure, lets the signal decay, lets them relax back in alignment with the bore of the magnet, and measures the signal. But what happens is you're measuring these different directions and you end up with little eddy currents. So just like you have a river, water's going down the stream, maybe there's a little rock here, and that rock causes a little bit of disturbance to the flow, and so then you can end up with eddies, these little whirlpools, and that's exactly what happens in the brain too. But the eddies are induced by applying the different gradients. Anyway, what happens is it causes distortions in the geometry of the brain, and that needs to be corrected as well. So what you do is you have your raw data, you calculate, you correct distortions, so you calculate distortions, you correct for them. There are different ways to do that. Then you fit some diffusion model at every voxel. That can be DTI, it can be some other methods. MOW is specific. It was developed here by some of my colleagues at the University of Florida, but there are other methods that are used. You can do tractography, reconstruct pathways, and compute different measures of anisotropy or these diffusion metrics and use that for the research. So really, first steps, and this is all done mostly automatically by software, is correcting these distortions before we do any of the calculations of metrics that we'll use.